Hello, everyone. Welcome to CyVerse's Spring 2023 webinars. I am Tina Lee, CyVerse's User Engagement Officer. We are honored today to host a webinar on BisQ, an open source data storage and analysis web service developed by the Vision Research Lab at UC Santa Barbara. And we will feature how the Smithsonian Institution is harnessing BisQ's AI image analysis capability to achieve some of its wildlife conservation goals. This is applied research at its best. Some of you may have used an earlier version of BisQ and CyVerse to do image segmentation or 2 or 3D object recognition, or to easily annotate your images. And this is the next generation of BisQ. So it's really exciting for me and our team at CyVerse to see what's new. Our presenters today include 3 collaborators from the BisQ-Smithsonian team: Connor Levenson and Satish Kumar of UC Santa Barbara's Vision Research Lab, and Lacey Hughey of the Smithsonian Institution. The principal investigator behind this collaborative project is Dr. B.S. Manjunath, who runs UC Santa Barbara's Center for Multimodal Big Data Science. Let's take care of some light housekeeping, and then we will dive right in. Today's webinar is about 30 minutes long, with plenty of time for your questions at the end. Please open the Zoom chat window and type your questions there for the presenters to address when they're done. This webinar is being recorded, and I will post the video recording on our website in about a day. And so now I'm pleased to introduce our first presenter, Satish Kumar, a graduate student researcher in Dr. Manjunath's Vision Research Lab. Welcome, Satish.
Oh, thank you so much, Tina, for the amazing introduction. Hello, everyone. I'm Satish Kumar. I'm a 5th-year PhD student at Vision Research Lab at UCSB. I've been working under Professor Manjunath since 2018, and I joined BisQ team in like a month before I joined my PhD program. So today we'll be going over overview of BisQ and how Smithsonian are using it. Okay, so the talk outline is as follows. So we'll start with what are the conservation plans that Smithsonian have, and Lacey will go over the problem statements and the challenges that they are facing with it. And then we'll move on to how BisQ is addressing those challenges and how Smithsonian is using the BisQ. We'll also show you how you can use BisQ for your data storage and visualization. And this will be followed by a live demo. And at the end, we'll highlight a new integration, a new feature of the BisQ in which you can play a video and you can do video analysis too. So I'll hand over to Lacey.
Thanks so much, Satish. Hi, everyone. I am an ecologist and a program manager at the Smithsonian National Zoo and Conservation Biology Institute. So I spend a lot of my time thinking about wildlife conservation and monitoring protected areas and trying to assess the health of ecosystems. So one of the most common ways that we go about this in the conservation field is by conducting wildlife surveys. And traditionally, these are done either from the ground, from field teams with vehicles on the ground, or in the air, because we're trying to cover really expansive areas. And so we're going to talk mostly about photographic aerial surveys and how the BisQ platform is helping us to automate the detection of animals in imagery. So I think the overarching challenge to any type of machine learning driven analysis of imagery is just the detection of small objects in complex environments. So you might not think of an elephant as being very small, but if you look at this image, I would tell you there are several elephants in here, and maybe some of you are seeing some, but if you're not, that's okay. We're going to zoom in and just show you who's in this image first. So next slide. Yeah. So there, maybe some of you saw this one that's kind of out in the open. Next slide. But there's also this cluster of 3 elephants in the bushes here. So we can do the next one. This is just to point out that detecting animals in these environments is already a challenge for humans. So it's it's going to be a challenge for machines as well. This is another fun example. So if you think you see something, keep your eye on it, and we're going to zoom in. So this is actually a group of zebra. Just around 10 zebra in this one. And next one, there's actually 2 wildebeest sleeping in the bush, under the bush in the lower right corner here. And so these are some of the more challenging applications that we are dealing with. But one more just for fun. There are some of the world's tallest animals in this image. So there's 3 giraffe down here in the lower left corner. So you can go to the next slide. Yeah. Our primary challenges are identifying small objects in complex images. Trying to improve, I guess, the fact that a lot of analysis of these images is currently done by humans. So people sitting in a lab counting animals by hand, which takes a lot of time and is also difficult to validate. And even in cases where there are machine learning models being published for detection of animals and imagery, these often are not very easily discovered or made very accessible to end users. So they're often buried in supplementary material of academic journals, or posted on GitHub. And a lot of times end users don't have the technical expertise to actually go find those, assess which model might be most appropriate for their particular application, and then actually apply them as well. And, oh no, there's something, can you click the, yeah, okay, I think the formatting got a little messed up on this one, but BisQ is really helping us solve these problems by, first of all, producing an image analysis and annotation platform that's cloud-based and open source, which is really important for our partners in areas without a lot of computing infrastructure. It keeps it low cost, keeps it accessible. And another really awesome feature right now is that you can actually mount your trained models into BisQ so that they're available in a graphical user interface for end users to apply to new images without needing to code actually at all. And then in our specific case, also having the ability to handle multiple file types. I think BisQ handles hundreds of file types, including GeoTIFFs and GeoJSONs and KMLs. So it can, which is really important for our work, which has a spatial component to it. So we often want to know where animals are on the landscape so that we can then analyze how they're responding to different environmental features or different changes in the landscape. You can do next slide. So to just sort of demonstrate the breadth of BisQ's abilities so far, we've started out with this problem with African megafauna and are moving into some of the smaller creatures. So we've got prairie dogs here on the left and black-footed ferrets on the right. There's a Smithsonian team based in the Great Plains in Montana that's trying to monitor the prairie dog population because prairie dogs are an important, they're considered a keystone species. So they really influence the health of the ecosystem around them in a way that's larger than many other species. They're also the primary food source for an endangered species, the black-footed ferret, which is these guys on the right with the little masks. And the Smithsonian is currently doing a reintroduction project where they're reintroducing black-footed ferrets on the landscape and wanting to monitor how the impacts of that on the prairie dog populations. So we're doing that by flying drones over prairie dog colonies and then using BisQ to automatically count prairie dogs. You could do next slide. So this is just a quick example of what prairie dog challenges. So and also the types of classes of objects that we're trying to identify. So there's prairie dogs in burrows, and then just the burrows themselves. And so you can pretty easily see the burrow. The prairie dog in this one is actually in the lower left corner of that burrow. But it's also a significant computer vision challenge, so that BisQ has been doing well with. I'm gonna stop here because Connor and Satish are gonna dive into the more technical side of how they're actually doing this. But thank you.
All right, thanks so much, Lacey. So as Lacey mentioned, like most of the challenges that comes around with working with any kind of machine learning module or data storage out there, either the data storage is sitting independent somewhere and you can't even interpret your data, or the machine learning modules are like sitting in a GitHub repo or somewhere in the paper, which are like difficult to reproduce by someone who have minimum expert. Even like me being in the field for 5 years, and we find it like sometimes challenging to even run a GitHub repo, like reproduce a GitHub repo, and you even reproduce the like results. And that's where BisQ comes like super handy for anyone. So it's like an open source web-based platform that supports data, like petabytes of data storage. And when I say data storage, it means like it's not just you can upload your data and that's it, it sits over there. It's way better than that. Like there are almost like 272 file formats that exist out there. And out of them, BisQ can interpret 250 plus of them. That means if you upload anything to BisQ, in 90% of the cases, BisQ will interpret it for you. You can visualize that inside BisQ, you can parse metadata separately, and then since like when you upload any kind of image into BisQ, it parses the metadata separately. And that actually make the query system super fast inside BisQ, even with you have like petabytes of data. And moving on to like the visualization part. So inside BisQ, you can visualize 2D, 3D, 4D, up to 5D images. It was initially designed for biomedical images where we used to deal with nifty or DICOM images. But now BisQ can support like all kind of GeoTIFF and GeoJSON file, and you can like add annotations to the file, and BisQ will separately keep all the annotations. You can turn them on and off. We'll show you the examples as we proceed further, how you can visualize annotations on an image, and how you can download the annotation separately or download the data separately too. And once you have both of these things together, we bring in the third most powerful tool like inside BisQ. That is, you can create your own modules. It's as simple as plug and play. Like you have your source code, and then we'll plug it inside BisQ, where you can share it with a wide community. Anyone can just run your reproduce your results or run analysis on their data set or your data set just with a click of a button. It's as simple as that. And everything runs from a web interface. Like I even tested it from my phone. Just imagine visualizing or running analysis on a 3D image just from your phone. It is like that powerful. So like I've been talking about BisQ, and Lacey also mentioned. So this is how somewhat like BisQ looks like. So as you go onto the login window, like BisQ.ec UCSB.edu, we are hosting it from like currently from UCSB, and we also had an instance running at CyVerse. You can access either one of them. So this is how BisQ looks like. And over here, let me enable my laser pointer. So it's actually easy to pointer options visible. Okay, I don't know how to enable that, but anyway. So over here, you can like as easy as we'll show you like how easy it is to upload a data. You just like a drag and drop. You can download your data in any format, and then the analyze button, it actually like drops down a whole list of like a library of machine learning modules or yours like that are integrated inside BisQ, and then you can run your analysis. And then browses, you can actually browse your whole data set. You're like by data set, you can browse by images, you can browse by file. And even if another edit thing that I forgot to mention is, even if BisQ doesn't understand your like data type, it will still keep that as a blob. That means it doesn't mean that you cannot upload it to BisQ. BisQ will, you can still upload it to it. BisQ will keep it as a blob, and you can actually export it in like as a blob only. Your data will stay intact over there. So as I mentioned, you can visualize. So here is the power of visualization. So the image that you see over here is not a regular image that we see on our phone. It's actually 100,000 cross 100,000 image. Like this image has been shared by Smithsonian group only, where they are working on detecting prairie dog, as Lacey mentioned, the same project. And you can see like how easy it is to annotate, like zoom into the image. BisQue is seamlessly parsing such a like large image, and how easy it is to annotate examples and edit those annotations. So that's what like a group at Smithsonian is working with us to actually annotate a large amount of data. And then we are training machine learning modules for them and integrating them inside BisQue. And then since I mentioned like how, like once the data is annotated, how you run analysis. So it is just a simple one-click analysis. You like from the analyze drop-down menu, as I mentioned before, you just select your module, what kind of analysis you want to do. So here we are showing an example of prairie dog, where you can run analysis on a whole data set, on a single image or a set of image. And as easy as like, and you hit run. And here BisQue is highly scalable. You can run analysis on the whole data set based on the requirement. It will scale up the job or scale down based on how much compute power it needs at the time. And our backend have very good GPU integrated inside BisQue. So over here is actually parallelly running analysis on a whole data set that we are showing. And then once those analyses are done, it is a very simple, like you can even visualize and edit those analyses too. So this is a whole data set on which like a tiny data set sample on which we ran the analysis. And you see, these are the few predictions by our model. So there is a prairie dog sitting next to a burrow, where you can like edit your annotation. If your model has done a good job, you can keep as it is. Or if you want like the annotations to be modified, you can move them around. You can resize the annotations too. And if there are like a few like mispredictions, so definitely machine learning modules make some mistake, you can actually like delete predictions, delete the annotations from your data set too. So, and everything is like is done through a web interface in a UI. And coming to the next part of it, that is where like a running analysis module. So that part also we have streamlined for you. So it's as simple as you ship your source code to us. It could be a GitHub repo, or you email your code to us. And then we have a predefined BisQue module creation template in which you import the BisQue API. So it can be even created through a Jupyter notebook. You can import like BisQue API inside a Jupyter notebook, and you can develop while your data is sitting on the BisQue and then access that. So you import the BisQue API and then some other dependent libraries that you need. You plug in your source code over there, and it can generate output in the form of an image, or it could be a matrix, or it could be a video output, or any other format that is needed. So once we have created that, like we have plugged into that module template, so we'll just Dockerize the code base, and this is how your module will show up. So over here, like in the right from the analyze dropdown, so this is like the prairie dog has been integrated inside BisQue. This is just a three-step process, and maximum it takes like a day or 2 days of time. And then you can share this with any number of users like you want. You can even like your analysis, like another thing that we ensured inside BisQue, data sharing and analysis sharing is very secure and very simple too. You can share your data with very specific set of users, or you can make your data set public. Same is with the module execution too. You can create groups, and that, okay, this tiny group can only use my module. For example, we are providing highly powerful GPU resources, and we don't want like anyone from outside to actually like make a wrong use of them. So you can actually set a very like constraint to use, like how people can use it, use those resources, and then who all can use it. So sharing is super secure and very easy inside BisQue. And once that module is created, so this is an example like on that created module. So how you just click on that drop down button. And then again, we are just showing like you just upload a single image, a set of image, or you can upload like image directly from your computer or a whole data set. And then as simple as you hit run, and then it will run the predictions for you. And another beautiful thing about BisQue is it keeps track of the history of executions that you have done on every single image. So let's say I ran a module today, and then Connor ran a module day after, and then Lacey ran like a week after. So BisQue will keep a track of all of them. And you can give a name of them. Okay, this is when, on what date that like Satish did that analysis or Lacey did that analysis. And you can compare them. Okay, like this is how like it did in first run or second run. And now, since I mentioned about the BisQue API, so we have integrated some external tool inside BisQue. that is a Jupyter notebook. You can develop a code, develop alongside BisQ using a Jupyter notebook. And also so in Jupyter notebook, it's very simple as you just import the BisQ API, and then you with your simple username and password, and you can import your data. And so your data is still sitting on the BisQ, and not you can't access everyone other's data. You just access your data. So you can load your data, and then you can do whatever type of analysis you want to do on the data, or if you if you are building a module, so you can actually load your data from BisQ and then build your module over here, all your code execution and visualize. And then I'll hand over to Connor, who will show a live demo of everything I mentioned.
Cool. Thank you very much, Satish. So now I'm going to give a live demonstration of some of this capabilities that were highlighted by Lacey and Satish. So as long as we can see my screen, okay. So here I am at bisq2.ec.ucsb.edu. This is a deployment of BisQ that we have here at UCSB. And this is where we manage data and develop modules, as well as deploy modules for our users. So I'm just going to navigate to the upload button here. And in my local storage on this computer, I have a few of the images that you guys saw from Satish's presentation. So I'm just going to select all of these images here, and then just drag and drop them into BisQ's upload UI. And from here, I'm going to click upload. And when I click on upload, you'll see that BisQ says that it is ingesting the data, which basically means is looking at the extension, it is parsing out the metadata, as well as uploading the raw image data. And for the sake of organization and for this demo, I'm going to add this all of these images that I just uploaded to a new data set. So maybe I will call this demo 2. And as soon as I add this to the new data set, we can navigate to the browse datasets page, or in this case, since it was the last one I uploaded, it will come up on my homepage. And I can actually click on it and go look at all of the images that I just uploaded. So this is the dataset viewer. There are a few different things that you can do in this view. You can set your data to be public if you wanted to make your data publicly available. You could share your data with specific BisQ users. You can add images to this particular data set if you want to add more images at a later date. And you can also visualize and annotate your images, which we'll give an example of now. So this is the annotator viewer. I could go here, I could go in and see any textual annotations, any graphical annotations, the metadata, and any past analysis that I have run on this particular image. And from this dataset viewer, I can run some of the modules that Satish highlighted inside of his presentation. So all I'm going to do is click on this Analyze button and navigate to the prairie dog detection module, which is what this data is specifically for. So this module runs an object detector on these images that I uploaded and outputs the bounding boxes where we predict prairie dogs and their corresponding burrows. So for example, if I were to fix these annotations after, maybe I want to call these new annotations that we will create the ground truth, and I will simply click run. And after I click run, I can see the status of the module being run on every single image in my data set. So this will take a little bit of time to run, but we will first see inside of our module that we developed, we leave some logging statements just to show exactly where things are at. So to run on this full data set, it will take a couple of minutes. So I have pre-computed this module on the exact same data set actually. And we can take a look at what the results will look like. So I will navigate back to the data set viewer. I will click on the data sets that I just ran my analysis on, and I will go back to the annotation viewer. And when I go back to the annotation viewer, I can see the output of our module actually on the images that we uploaded. So I can go here, I can go to the graphical annotations. I can see this is what I named my annotations, and I can navigate through all of the annotations that were created by the module. And then from here, you could do everything else that you could do with the Bisq UI annotator. You could move these annotations, resize them, rename them. You could add new annotations if it was missing, or maybe if this wasn't a burrow in this case, I could delete it. And then from here, I can navigate through each of my images in my data set and fix those annotations one by one on each of the images. And then once I am done fixing all of the annotations that were created by that module, I can simply go here to download, and I can download the graphical annotations in the corresponding format. So in this case, I could download it as XML, and this is kind of a lot. You don't have to parse all of this, but you could see for each image in my data set, I could see, okay, here there's a burrow, and these are the coordinates of the burrow inside of that image. So that is an example of one of the modules that we have developed here. I'm going to give an example of another project that we have worked on for some marine science researchers. And this is doing video analysis on underwater video. So in this video, as you can see here, we are interested in counting the number of underwater species that occur. So this species, if you can see it based on my cursor, is called a fragile pink urchin. And this is a species that the researchers are interested in actually counting. So they have like hundreds of videos. And what they want to do is just go through all the videos and count how many of these species occur in all of the videos. And so we have a species tracker and detector module, which takes as input videos just like this. And so very similarly to the last module, you just navigate to the analyze, you select what module you like to run, and from there you can just click on run. And this module, since it's running on a video, it does take a bit of time as well, but I have pre-computed a module on this specific video before, just like the last one, and have some of the outputs. So here I can go to the browse and go to the module execution browser. And here I can see all of the analysis that I have run for this specific Bisq account. And I will go to the module that I pre-computed for this demo. And Bisq modules are pretty agnostic to your output. So you can have your output be whatever you want. So in this case, you see we have three outputs here. We have a table, which shows us the count of the species in this particular video. So we navigate here and we see, okay, 17 fragile pink urchins were detected. We have a few different annotation formats, which are compatible with different annotation tools, which I will show. And we also have the output video, sort of visualizing exactly what is happening in this module. So if anyone has ever annotated this type of data before, if you have videos of some sort of species, you know that you sort of have to have a strict definition on when you start to count things. So in this case, we count the objects in the video if they hit this line. So we have this sort of remote vehicle going across the bottom of the ocean. And if the species hits this line in the middle of the video, we count that species. And this is just a visualization of that happening here. So next, what I'm going to do is show how we can actually have the outputs of our module be compatible with external tools. So there's something called CVAT, which is a computer vision annotation tool. And the output of this module outputs an annotation file, which is compatible with CVAT. So I will just navigate to this annotation file. I will download it, and I will navigate to the same video, but this time on a hosted version of CVAT that we have. And this is the same exact video as I upload to Bisq, but it is sitting on CVAT. And I will upload the annotations that I just created from that module. Just like so. And as long as everything goes fine, we will see the annotations pop up here shortly. And so the output of this module is actually something called a CVAT track, which basically adds some temporal continuity to the annotations. So we will see actually as we step through the video the same species will be tracked. And obviously this module missed a particular species. So we can use CVAT in this case to actually add a new annotation of a fragile pink urchin in a new track and fix those annotations. And then from there, I can use CVAT to do whatever I want in CVAT. So I could add more annotations, I could export the annotations, and then obviously visualize them. So yeah, so that's it for our demonstration of BisQ. I will now pass it back to Satish, who will wrap things up, and then we can take any questions.
All right. Thanks so much, Connor. That was actually pretty cool. I was a little scared and like, the standard problems with the demo. So things actually falls apart only at the demo time. So, but yeah. So, oh wait. Okay. So after the demo, so we have a number of users who have been using BisQ. The biggest users are CyVerse, where CyVerse was hosting a BisQ instance. We have like approximately 35,000 users. And then Smithsonian has been extensively using BisQ over the last couple of months for data annotations on like 2 different projects and also running analysis. One is the prairie dog, and another one we have also been working with videos of like arc detection from camera collar videos. And then in University of California, Irvine, the Neurological Surgery School of Medicine have been using BisQ for annotating and running inference on brain images, where they actually draws like a segmentation mask on like certain regions of the brain to detect some disease called like NPH. So that's all from us. So our BisQ team is like few of the members are missing today, but you can actually scan this QR code to actually try out BisQ, which is at the bottom left corner. And you can actually also look at our GitHub repo, where you can also have your local instance of BisQ. You need not to use our BisQ. You can actually host it from your own computer. It's like pretty simple to deploy even BisQ on your end too. And this project has been successfully running for last almost 15 years by the support of NSF. And we are very thankful for that. And we are open to questions now.
Thank you, Satish and Connor and Lacey. I don't see any questions at the moment, but actually I have a question. Can you tell me, maybe Lacey could start, how this collaboration came about? How did somebody at the Smithsonian find BisQ, the BisQ people?
Well, yeah, something I skipped over was that I did my graduate work at UC Santa Barbara. So I started some of this work trying to detect animals actually in satellite imagery. Was how I started out working with Manjunath's group under Doug McCauley at UCSB, and have carried that with me into my career. Here I'm working for Smithsonian because they had lots of similar questions and applications in terms of wanting to monitor large landscapes from remote imagery.
Thank you. Okay. I still don't see any questions. So my one other question is in terms of security of the information. Endangered species locations are probably not available to maybe poachers via this app, but is there security built into BisQ for things like either health data or sensitive data like tribal data or endangered species?
Oh, so that's actually a pretty good question. So as I mentioned, like our data, data sharing inside BisQ is very secure. It is going to be public only if you make it public. And then you can make your data and annotation separately public. Like you can just let's say open source your data so that anyone can use it. And then you can actually hold back your annotations where all detections actually happen. So, and then you can share that with only a specific set of users that you want with. And if no one else wants to use, then it will stay with you. Even we as like a BisQ admins, like we can't even access that data too. So it's actually specifically tied to the user who wants the data.
Okay, thanks. There's a question. I'm curious, would there be a minimum image quality or resolution that is required or recommended for BisQ?
Oh, I can take this one. So there is no minimum resolution that is required for BisQ. BisQ has got nothing to do with it. It totally depends on the given image, what you are detecting, and also like whatever algorithm you have built for detecting that particular object is actually able to detect that or not. We just integrate that inside BisQ. And if you want to use some pre-built or pre-integrated modules inside BisQ, then if you're we might have to fine tune the model to actually do those detection. Let's say we have very poor resolution on the objects.
I would also add for the wildlife detection case, at least with our early models, like we were using YOLO 3 in the beginning. Connor and Satish have been playing around with newer versions of YOLO and other models, so you may be able to get away with less than this, but we usually aimed for about 20 to 30 pixels per object that we're trying to detect, in case that's helpful.
I think that would be helpful. Another question, is there a getting started with BisQueAI file for new users to learn how to navigate the software?
Yes.
Documentation, yeah.
Yes, we have a detailed documentation if you go to our GitHub repo, where it actually step-by-steps walk you through like how to use BisQ and also how you can even set up your own local instance on BisQ. And even if you go to our bisqec.ucsb.edu, on the like top right corner, we have a YouTube link to where it shows like how to use BisQ. Like there's a YouTube tutorial also.
Okay, great. Another question, would it be possible to use BisQ to just share the results after analysis with a different team, even if the data is private?
Yeah, I think that's possible. Thank you, Shalja. So let me make sure I'm getting the question correct. So let's say you were to have some sort of let's say output segmentation, but you don't want to release the like private data. So yes, you would be able to do that because the annotations would be new data on BisQ, and you would be able to share those while keeping the original uploaded data private.
That makes sense.
That answers the
Okay.
question.
One more question, can Google satellite images be used in BisQ?
So does this mean that you want to directly import the satellite image inside a BisQ? So if it means that, then I would say no, right now, like you cannot directly import Google satellite image. But if you download them and then upload them to BisQ, and then you actually want to do some analysis, for that, actually, I have some examples. I used images from Sentinel-2 satellite and also like airplane images, like hyperspectral images. And over there, you can visualize them even like I tested even like up to like your normal RGB image have like 3 channels. I tested up to like 432 channel, and BisQ can actually interpret it for you. So I hope that answers your question in both the cases.
And I'll add one more thing there too. If you're talking about Google Earth imagery, that, yeah, as Satish mentioned, the biggest issue there is just the legal requirements. So usually you can't actually download those images directly, but if you do have access to satellite high resolution or whatever resolution images you're trying to get, you can work with those. That was actually the first data sets. We used GeoEye and worldview data in BisQ during my dissertation. So, and it worked. It handled those large images amazingly well. So basically GeoTIFFs, and also, as Satish mentioned, multiple bands that you can visualize the bands in there. So I was really impressed. Even it was also just really helpful for sharing, for collaboratively annotating those images. So it avoids having to hand out copies of your satellite image to multiple users to view in QGIS or whatever platform you're using. You can all collectively annotate at the same time, and it handled it really well.
Thanks, Lacey. Okay, and I did anticipate that somebody would ask something like this. Can ChatGPT be used, or can ChatGPT use the public data from BisQ?
So I would answer this question. Like ChatGPT only interprets textual information, and we don't have textual information inside BisQ. But yeah, if like the annotations inside BisQ, they are in the format of JSON, and that's or XML. That is basically text. So ChatGPT actually can use that kind of information from BisQ. You can like download it and dump it to ChatGPT and test it out. Like, how does it interpret that?
I would say too, I don't know if this is exactly what the question is asking. If I can go back to a different slide, possibly, but you could combine the different APIs. So in this case, right, let's say you did have text data stored on BisQ, or you had some sort of multimodal version of ChatGPT, which could interpret images as well. Then you could just use the BisQ API as well as like OpenAI's API and build a program which, gets the data and can even get analysis from BisQ, and then could use something from OpenAI or any other API that you want to do further analysis on the data.
Lots of flexibility there. Okay, with no further questions, thank you, BisQ and Smithsonian collaborators, for sharing this. I'll use it and just be yucky, this visionary project, which highlights the best of what applied AI can do, computational research and tools and collaboration. If you've signed up for our next CyVerse webinar on cloud automation and continuous analysis orchestration, we'll be rescheduling that due to a staff conflict, so stay tuned for a new date. Other than that, thank you everyone, presenters and attendees, for coming to our webinar. And Connor, Satish, and Lacey, there's a lot of thanks in the meeting chat for you. So see you next time.
Thank you. And thank you to everyone who attended.
Thank you.